It's 11 o'clock, so I think we should get going. We have 7 panelists ready to sort of steer discussion and sort of work together with the attendees. We have 32 attendees, and that could probably change over time as well. So I wanted to start off with saying a very warm welcome. It's very exciting to do this in this format. It's something that at least I haven't tried at this scale before. As you will know through our email communication, we made the difficult, but we feel right decision last week to postpone the ICDA face-to-face meeting with the acknowledgement that a lot of what we need to do in terms of setting up the group and meeting face-to-face and discussing needs to still happen. But at this time, it feels like it's better to postpone and cancel and do it virtually given the current coronavirus outbreak. So we want to thank you for all your patience and all your forthcoming and all your positive feedback around this decision. So that felt very encouraging because of course it would have been much nicer to see you all in person. So we're trialing this format today, and I don't know about you guys, but I'm at least a little apprehensive to see how the discussion will flow and so forth, but I'm also excited because it might be something that we need to do over the coming weeks or even months. So together with you and your input, we will make it a success. As you know, we will have 2 town halls today, and that's in order to try to accommodate time zone differences. So I've seen we have a couple of call-ins from Australia and other places, which is great because that was our intent. So Loic, I've seen you there, welcome. And then I also wanted to talk through quickly the Zoom etiquette, which Rachel and her team has sent out in the communication. So if you look at the bottom there, you can raise your hand if you want to say something, and you can also use the chat page at the bottom here to suggest comments. And for instance, Rachel has just told me I should remind everyone that you can ask questions and upvote questions during the Q&A that is coming later. So you will have seen the agenda. When I quiet down here, Eric is going to kick off and give you some general overview of ICDA. Then we're going to go in and discuss through the different recommendations, and then we're going to have panelist sort of discussion that Caroline and also to a lesser extent me will sort of moderate, and at that point we will welcome your questions and input as well. The link to the recommendation has been sent in the agenda. I'm happy to sort of put it in here as well if anybody hasn't seen it. So I also want to extend a big thank you to Rachel and her team for sort of putting this format together and for very quickly going from a fully organized and ready conference to go in Copenhagen with kind hosting of Novo Nordisk, who's also been super helpful, and Søren and his team to sort of swiftly going over to this virtual format. So thank you for that. So without further ado, Eric.
Oh,
here we go. Now you're on. Now you're on. Yeah.
Yeah. Thank you, Cecilia. And let me add my thanks to Cecilia's for all of the work that was done to set up Copenhagen and then unwind Copenhagen and set this up on very, very short notice. And thank you to everybody who's on the phone. We now have 79 people on the phone, which is fantastic. And one advantage of this format is I know we have a number of people joining who were not able to go to Copenhagen, and that's great. So let me just say a quick word by way of framing before we're going to turn to lightning overviews of the recommendations and then to general discussion. What is ICDA? Still worth emphasizing what ICDA is and what it isn't. ICDA is a scientific forum to bring together people interested in this maps to mechanisms to medicine challenge from all around the world, many, many different countries, many sectors, academia and industry and government, funders, and many, many more, to try to identify what are the most important things we need to do together to solve this M to M to M challenge. to help facilitate that those things will get done, and also to help convene the community to sort of monitor the scientific progress. But what ICDA is ICDA is not going to do the science itself. It's not actually in charge of anything. ICDA doesn't decide what gets done or not. That's going to be up to the scientific groups, to the funders, to decide what they want to do, what they want to fund. But the act of bringing together an amazing community that hasn't really had a formal channel to try to distill the ideas and clarify the projects, I think the work that's been done in the last 3 or 4 months by working groups has shown how powerful it is to bring together a community to try to reach clarity about what needs to be done. The purpose of this call is to look at those recommendations. I know these recommendations, they're not prioritized as this is the one to do first or the other, because different groups may wish to do different things, and different funders may wish to fund different things. So for all those reasons, the idea is to lay out what are all the things that are going to have to get done. I think ICDA and all its members will have a very useful role in talking amongst the groups, talking to funders, to help people understand what it would mean to pick up one of those recommendations and run with it. And it might be that several groups together, as is likely going to be the case, want to run with things. And I should emphasize that in many cases, groups are already doing some of this, and it's likely that they'll be the ones who are running with it. I think very little would be on the list if it was a total vacuum. Indeed, the recommendations grow out of work that's beginning throughout the field. So ICDA is meant to be able to help facilitate those discussions. And then I think as things continue to grow under perhaps this intellectual framework, to bring people together for scientific meetings to talk about how it's going. So that's just by way of background, so people know what we're trying to do. We're not in charge of anything at all, and yet a community that takes the responsibility together to clarify its goals is a very, very effective community. So I'll stop there, and I think our plan is now to go into lightning, and I think it really is lightning descriptions of recommendation clusters, 2 or 3 minutes, so that we can open up. Everyone has got the recommendations and has seen them. They've been available for at least a month. So we're not going to go into great detail. But anyway, let me thank you everybody for joining. 101 people so far on the phone. Back to you, Cecilia.
Great. Yep, I'm on. So then I think we're going to go back to the agenda, and I actually think it's back to us, Eric, for recommendation 1, where we're going to discuss through the flagship project. So just to add one thing to what Eric said, I think it's really important also that everyone who is active in ICDA can, of course, also stand up projects. Because that's something we have discussed, that a lot of scientists that are active in ICDA will stand up projects. So one of the things that we are going to do now is that we're quickly going to do the lightning thing, where we go through the different recommendations. Eric and I have been assigned to go through the flagship disease projects. And for those of you who read the recommendations, you will have seen that we have proposed that a collection of flagship disease projects will act as a test bed, or like a pipeline construction to develop and validate reliable paradigms for how to go from maps to mechanisms to medicines for any common disease. Of course, it will be cases in that sort of flow that are special for each of the diseases, which tissue type, which cell type, particular mechanisms that will be mapped out. But we could think that it's a reasonable thought that there will be sort of a flow that we will need to sort of map out. We need to identify the causal variants in genes. We need to figure out the cell types and tissues and organs where those genes and variants act to affect disease. We need to map out the cellular pathways and mechanisms that drive disease. we need to identify potential targets for prevention, detection, and therapy, and any kind of tools that are needed for diagnostics and therapeutic development. So this is obviously really, really, you know, a grand sort of stage, if you want. And I personally love the executive figure. If you look in the recommendations, the overview, where you will see that the flagship sort of projects are the ones that are cross-cutting the sort of three themes. And I think that that's sort of on purpose, because again, it will allow us to flow. Eric, do you want to add something to my brain dump?
Yes, let me just briefly say, there's of course already work going on many diseases, and this is intended to kind of provide a framework under which many such projects go along, where we can think about what are shared infrastructures across them. What if some funder or disease area wants to go into new areas? It tries to think about what are the common things that one would do across diseases. These things will flow from existing projects for sure, but I suspect will grow. Some people have said to me already, who decides what should be a flagship? The answer is the people who want to do it and the funders who want to do it will ultimately decide that they want to do it. It's not like ICDA will decide what should be, you know, what the choices should be, but ICDA can help clearly define what would constitute a meaningful flagship project and help ask what might be the most efficient ways to stand it up. Those are the kinds of things that you know, we want to have this for. And it was recommendation 1 because it's cross-cutting. If we can do recommendation 1, it becomes the forcing function that tells us we really can make use of all the rest of the pieces to understand disease.
And I think it's really important the champion part, because somebody has asked us, how would we work with existing disease consortia, for instance? So I think we have no intention in competing. It would make sense that you know, somebody in an existing or all of an existing disease consortia would stand up a flagship process and sort of map it out. I think there would be merit in, you know, doing an example sort of flagship project and sort of map out roughly what that could look like to sort of help people guide through that thought. But I think it needs a champion, and it needs to then go to funders for discussion. Does that make sense to people?
Actually, we're not getting feedback at this point. We're doing lightning everything.
No, I meant to you, Eric.
Sorry. To me. So let's let others go through the lightning for the first set of clusters, and then we'll be able to be open to you know, getting the questions that have already come in and that are appearing on your Q&A window here.
Perfect. Okay. So if we're done with a flagship lightning, we should go on to Mark Daly, Ben Neal, and Kristen Willer about the MAPS recommendation. And I think it's Mark, who's going to go through this in the first session, right?
Yes, exactly. And as long as nobody has muted me and I can be heard, I am happy to do so. Okay, a thumbs up means I guess people can hear me. So the MAPS group, very ably chaired by Ben Neal and Kristen Willer, has been focused on, obviously, the first step in this pipeline, which is what are the primarily resources that are needed in order for us to develop a comprehensive and fully transparent for the rest of the research community view of the genetics of human disease and genetic variation in general. And so we ended up with a few focus areas with respect to MAPS, and then a few others that I'll highlight with respect to the genetic resources that are required. So number 2 on your tour through MAPS, the Mechanism to Medicine, is quite obviously the critical importance of increasing population diversity of biobanks that are focused on genetic analysis. And I don't think, I'm not going to go through the justification for this. I think it's obvious to almost everyone, and probably at least almost 120 out of 120 people who are now on the call, which is fantastic. But the specific activities is that, that the ICDA recommends, are, of course, that funders need to really focus on emphasis facilitation of supporting the construction of biobanks in underrepresented regions of the world, as well as including underrepresented populations more fluidly in existing biobanks, and working with the regional scientific communities to make sure that that happens in all locations in the world. The next point, point 3 on your map, is that we would need, sort of in sync with that, to increase globally and very substantially the size and utility of biobanks focused on genetic analysis, with really an ultimate aim of working towards perhaps something on the order of 50 million individuals with a global diverse representation, but with a deep clinical and genomic workup of those individuals as a resource for discovery and all of the downstream things that we're going to talk about. We would hope that that would, in most cases or in many cases, be clinical data from primary care, from EMRs, in order to get as complete a real-world picture of health as possible. And that the funders should be supporting pilot projects in these areas to help design and evaluate and expand existing biobank efforts all over the world. And finally, recommendation 4 would be to support genetic analysis across biobanks, and not by aggregating in one place all the world's data as some sort of Uber project, but the idea that has emerged rather organically over the last year is that there should be a genetic analysis network that fluidly allows collaboration at whatever level it's appropriate for data sharing and taking advantage of advances in data sharing and data security, but that funders should really support efforts to create such a network, to facilitate its meeting, facilitate federated analyses, and perhaps even with respect to an early opportunity that a global biobank network has begun, that Ben, Kristen, Cecilia, and many others are very much involved in. And so hoping to support some of those meetings and activities. So the next section, section 3 or the second half of the maps, speaks specifically to genetic resources that are essential. So in addition to increasing the size and interactivity among biobanks, there are comprehensive resources that provide a critical starting point to all of these activities that we're going to discuss today and for the next couple of years. One is the need for a comprehensive genetic and genomic variation resource. It cannot be understated how important it is that we have as complete a picture of human genetic variation across all populations as possible. This needs to be comprehensive. It needs to be emphasizing especially underrepresented populations, as in the case of the increasing biobanks. And we need to ensure as much as possible that there's broad and individual level sharing when that's possible, but also that there are allele frequency servers and imputation servers and other ways of representing non-individual level data that, as many of us have come to know over the last decade, are becoming critical elements of both clinical and research discovery in common and rare disease on a day in and day out basis. And so those foundational resources and servers and tools need to be supported. And then finally, point 6, in a similar vein, one of the motivating things that brought us to the table in terms of an ICDA has really been this observation of great progress in one sense in the discovery of genetic variants associated to disease, common disease, especially via genome-wide association studies. And that has to be the launching point for all of the downstream efforts to better understand the mechanisms of disease and ultimately develop new medicines. And so as such, it's essential that we have and significantly invest in the resources to accurately capture and curate all of the genome-wide association data that has been collected over the past decade and a half of early and now more mature GWAS studies. These need to not only collect primary data from individual studies, but also work very closely with the most sort of detailed consortium efforts that have taken place in many different diseases in order to represent the best powered and most current up-to-date information, and to keep that up-to-date and in real time for the downstream mechanism and medicine communities that will build from this, as well as potentially develop other, as in the case of the allele frequency and genomic variation resources, other references that might be useful, for example, with polygenic risk scores. And obviously, extensive discussion is already taking place with respect to the EBI and the GWAS catalog that they have initiated. So I will turn over to continue the section, and on behalf of those of us here in Helsinki at FIM and my colleagues who have been involved in presenting at previous ICDA meetings, nice to see everyone.
Great, thank you, Mark. So we're going to go over to Tuuli Lappalainen, who's going to discuss through recommendations 7 to 9 in a lightning way, which represents the mechanisms session.
Okay, thanks, Cecilia. Hi, everyone. So yeah, it's a pleasure to present on behalf of the Mechanisms Working Group that is co-chaired by myself and Chase Nduri, and Chase in the middle of the COVID-19 work in Seattle and not able to attend today, unfortunately. So as you all know, one of the key challenges in common disease research is understanding the mechanisms, how these variant associations actually contribute to complex phenotypes. And here in the Mechanisms Working Group, we're really focusing on the molecular and cellular mechanisms, and then the physiological mechanisms are in the medicines part. And we put together 3 specific recommendations to address different aspects of this challenge. And this, of course, doesn't include everything that needs to be done in this area, but these are some focus areas that we identified. So recommendation 7, which is creating a human gene regulation map, really focuses on non-coding cis regulatory elements and variants and better identification and characterization of those. So here the idea is really focus on analysis across all human cell types, really cell type, potentially cell state specific analysis to identify all cis regulatory elements, a little bit sort of alongside of what ENCODE has been doing. Also identify all cis-QTLs at the single cell level, at the above a certain effect size threshold. Infer target genes affected by these cis-regulatory elements based on experimental data, CRISPR perturbations, etc. And then also develop computational models that use this experimental data to allow these kinds of inferences even when we don't have that data from experiments. And then also do sort of a functional inference, understand the functional architecture of these cis-regulatory elements so that we can actually predict or develop predictive models of variant effects on gene function and gene regulation in cis. And this recommendation, as some of the others, really links closely to some of the existing projects where we will need to interface with them to continue and complement their work, especially ENCODE and the Human Cell Atlas. Then when it comes to recommendation 8, this focuses on protein coding variants, which is of course a smaller slice of all the GWAS variants, but in some ways a lower hanging fruit in terms of understanding their mechanisms, because the target gene is known and the effect sizes are larger, and also coding variants it's a tractable number of total variants, which allows comprehensive analysis. So here the idea is to really sort of like use all possible approaches and ways to try to understand these variants, ranging from PheWAS analysis in various biobanks to various experimental perturbation approaches using isogenic genome editing, high throughput saturation mutagenesis, also model organism work for selected coding variants, and then various general functional assays or gene-specific functional assays. And then also a key part is to develop and calibrate improved computational methods to predict coding variant effects on protein structure and function. And then finally, recommendation 9 is sort of taking these kind of effects in cysts to an effect on cellular programs, which is potentially a more challenging sort of aim that we have, but something that is absolutely essential from getting from those molecular functions to actual cellular phenotypes, which is of course kind of an intermediate to getting to physiological phenotypes. And here, the recommendations that we have put forward are basically twofold. So there is one that takes advantage of existing genetic variation across individuals. So using samples from actual human individuals or cell lines, organoids created from those, analyzing how common variants or rare variants or polygenic risk scores associate to cellular phenotypes, such as sort of single cell level gene expression patterns and other cellular phenotypes. And then also experimental perturbation approaches such as CRISPR-I to inhibit genes and then have readouts on the consequences of that. And these would be parallel complementary approaches. And I think I will end there. I will just mention that in all of these studies where the idea is to use samples from existing human populations and cell lines, there's going to be a lot of work also to develop protocols and SOPs for creation of biospecimens and cell lines and assays. And then when it comes to population samples from humans, a key focus area is to have samples from diverse ancestries, to avoid the European focus that we have had in these studies thus far.
Great, thank you, Tuli. So next we have the data cluster, cluster 4, which are recommendations 10 to 12, where Ben Neal is.
Cecilia, this is Carolyn. I'm going to cut you off because I think, correct me if
yes,
the plan is to pause now for discussions because we knew it was a lot of information. So we're going to go ask now. This is Carolyn Hutter from the National Human Genome Research Institute in the US. And we're going to take you into what is now our first of 2 Q&A sessions, after which Cecilia will take us to cluster 4. So just so people know, what we're really looking for right now is your input and thoughts on these recommendations. If you don't see it in the bottom of your screen, you should see something that says Q&A, and if you click on that, you can enter in your questions. You can also upvote other people's questions and feedback. With 127 participants, we're not going to sort of unmute everybody, but we may try to unmute some of you as appropriate to bring in different questions. And what we're looking for this is, at the highest level, did we get these recommendations right? What do people see as sort of major strengths or gaps or questions that you have as you think about the current recommendations and as you think about what ICDA is doing or shouldn't be doing? You can also ask questions more generally about ICDA itself and not just the recommendations, if that's one of the places that you're wanting to sort of get some more feedback. So I'm going to sort of kick us off. I'm incorporating some of the questions coming in as well as questions coming from people who put some stuff in last night. And I would say it's interesting sometimes to see the overlap in questions. So I'm going to start with one that sort of was something that I had incorporating with Paul Franks, which is ICDA really has a very ambitious set of recommendations that cover a lot of the areas on this maps to mechanisms to medicine spectrum. And what are the thoughts about, or the framework for partnering with, identifying, integrating, and working with overlapping stakeholders? Examples include epidemiologic cohorts, existing efforts with the same aims. Paul called out disease-specific international precision medicine initiatives. Somebody last in the online talked about open targets, et cetera. And so maybe Eric, you lean forward, so we'll start with you. And this is sort of a question to any of the panelists who want to jump in.
Sure. So I want to emphasize again, I mentioned it briefly, but it's really important to say that we expect that most of this work is going to be done by groups that either already exist or are standing themselves up. So it's more like, how can ICDA help bring together and convene? But it's not, in many questions, the work may be an expansion of what is going on by existing groups. I think ICDA is trying to describe what needs to be done, but I know that in many cases there are very active, say, disease-focused groups, and it may be that they are providing the leadership for it there. In other cases, I know there are diseases that haven't been really the subject of great genetic focus, and they're trying to figure out, how do we mount a project? And I think ICDA can be helpful to such groups or to funders who want to see work in an area in helping describe what it would take to do that. But in the end, every one of these recommendations is going to come from initiative in the community and people taking leadership. ICDA can help facilitate, but it's not like ICDA can decide or can do the doing. We can act as a forum for groups together to do it. So I think ICDA is prepared to work hard with existing groups to help make connections, help make sure that there's access to infrastructure and tools, help make sure that the plans are fully fleshed out. But again, this is, it's going to depend on the initiative of the community.
Thanks. Do any of the other, Mark, did you want to add to that?
Yeah, I'd expand on it and also take David's question as well. I think, one of the key things to keep in mind is that, in response to both of these questions, is that as we think about flagship diseases, I think it becomes more obvious that that we can be reminded that the focus is really not on the genetic discovery activity, and the focus of collecting the largest possible biobank is primarily not for genetic discovery purposes for common diseases where genetic discovery is well underway. I think it's clear that flagship projects would be quite naturally led by, if they overlap with a significant consortium activity already going with gene discovery, then the gene discovery aspect of that would clearly be driven or have a large contribution from those consortia. I think what we are trying to focus on is really that we aim that we can now see through in these flagship projects initially the trajectory all the way from the mapping process, which is well underway, to an understanding of the mechanisms underlying what we're mapping, and then all the way through to therapeutic hypotheses. And so the biobank expansion, which as we do say in your final point, David, that yeah, the aim would be 50 million in a more cohort style biobank with the broad data, would make the possibility that the required biospecimens, the important understanding of the full range of medical consequences, positive and negative, for individual associations and for the global risk of disease, would be well understood. And so far more than just the discovery activity, I think we really want to focus on what is the full set of deliverables from genetics, and then do we have not only the information but also the biospecimens and the follow-up possibility to carry that forward.
Thank you, Mark. And Mark, just in the future, if you're referring to questions like you just did for David's, can you also let the group know a little bit about what the question is? Because I'm not sure that everybody can sort of fully see the questions, particularly people who just dialed in and didn't use the WebEx. So the
Great point. Okay, sorry, I have the Q&A thing up on my screen right next to my Zoom window, so I was presuming others did as well.
I think that
I will keep the questions.
Go ahead. So another question that's come in that's been upvoted quite a bit is from Mark Neslowski, and a huge apology to everybody and anyone whose names I mispronounce. But the question really sort of addresses phenotypic harmonization. So even when existing biobanks are fully digital, the choice of variables may not be standardized, but could be converted and adapted, which requires a deep knowledge on the phenotypes. And so I wondered if somebody wanted to talk a little bit about both phenotypic harmonization, and I'm going to expand this a little bit to also talk about what types of phenotypes are really are we thinking about and are recommending within the ICDA to bring in, and what types of things. In some ways, there's just sort of a mention of phenotypes, sometimes clinical, sometimes hinting at environmental, but not fully spelled out in the current recommendations. Where do we sort of see things landing for that? And I guess I'll knock this over, although I see he just took a bite, to Mark or anyone from the MAPS group who wants to take an answer to that one first.
Happy to take a shot, though. I mean, I think this is an area where there clearly needs to be investment, and then there's not a quick answer to this. I think in the proposal that we need to invest in the development of biobanks, this is certainly an essential part of developing biobanks, which is to figure out what is it possible to collect and how would we harmonize it with the remainder of the global activity. But it's not one that I think anyone has a quick or straightforward answer to at this point.
I would completely agree with that. I mean, I think it's worth noting in recommendation 4, we talk about both clinical phenotypes and other phenotypes, as well as, kind of explore phenotypes that have more traditionally been very difficult to capture, particularly things along the lines of progression and outcomes that may be more available from electronic health records and integration along those lines. But again, it is exactly as Mark says, that it's not as if we have all of these phenotypes packaged and ready to go. A lot of the work will be on the curation and harmonization on the phenotype side, alongside with the curation and harmonization on the genotype side.
Can I jump in for a quick second here as well? I think it's really important to emphasize that having as many phenotypes as possible, including medical record phenotypes, but also environmental exposures, to the extent that those can be obtained, self-report. I think the UK Biobank has shown the tremendous power of collecting a diversity of phenotypes. So we could probably we probably emphasize even more in the recommendations the importance of seizing the opportunities to collect phenotypes broadly. The second point I'd make, and I look to things that Mark and Ben have done, is that there are both traditional ways of harmonizing phenotypes by making sure that one collects exactly the same thing. But I think the ICDA community can be a leader in also thinking about modern ways, like using machine learning. There have been recent papers where phenotypes collected in totally different hospitals got integrated by machine learning to figure out when somebody in hospital A says something and somebody in hospital B says something else, this is what they mean together. And then finally, there's genetics, which is there's a validation that only genetics can provide, that if in one country a disease was defined one way and in another country a disease was defined another way, by looking at which loci come up as significantly associated, if they show the same sets of loci, then there's a strong case that they're detecting the same thing. Not every last locus, but broadly so. So I think the ICDA community working together could be very creative about the multiple different ways of harmonizing phenotypes that are now possible given big data and given genetics. And I think we should certainly, if we're looking to the future, be collecting phenotypes broadly, or more precisely, urging the projects that are stood up to collect broadly.
So yeah, and I think, to that note, I also think it's important with some of these things to think about what needs to be collected and thought about at the beginning versus what things can be done more retrospectively. You can think about sort of some things that passive collection could do, or you could go in and later get the information. And then there's other key non-genetic information that it's really good to think about from the beginning and engaging with the correct communities to think about how to be getting that data. I want to switch now. There's a question about how online combined with one of the questions that came in last night. So Ines Barizo was asking about how to ensure sufficient engagement with the LC community, particularly thinking about diverse ancestries that have been less represented thus far. How do you make sure this is coming in and working in efforts with the local communities? And although not exactly in the same vein, but also I think related to this question, is a little bit about what does ICDA mean when you say diverse, and how do you, sometimes it's unclear in the recommendations when you're focusing only on ancestry diverse or more broadly on other activities such as socioeconomic diversity, et cetera. I realize there's more to those two things. And Nikki did point out that this is going to be covered in the Cluster 6 recommendations. I saw that a little too late, but I'm going to go ahead and, since I already asked it, put that question out now. And I don't know, Nikki, if you want to chime in a little bit first.
Sure, no problem. Thank you, Carolyn. Yeah, we will be talking about that under our recommendations, which are around the developing good policies for genetic studies, data sharing, and federated analyses, and then also promoting global equity. And in these recommendations, we talk to our consent processes and how we can make sure these are sound, and also how we can accurately store the consent data, because often these are not really properly captured currently, especially for secondary data analysis. And then we also looked at under policies for sharing data sets, etc., that we need to develop community engagement guidelines that are regional and local, relevant regionally and locally, to ensure that the policies are inclusive, and also something that is not often flagged, but to address culturally accepted research practices. So not all cultural views of cell lines, in vivo models, bloods, etc. would be the same. So I will be speaking to this a little bit more. Perhaps we can, and equally with promoting global equity, to ensure that approaches are globally inclusive for participants and researchers. So I will be speaking to this a bit more in the next summary of recommendations. I think maybe we can pick it up there if that suits everyone.
Sounds good. Thank you, Nikki. So the next question comes from Carl Anderson, both online on the online submission and here in the chat, about why is the recommendation focusing on whole genome sequencing rather than whole exome sequencing? Eric?
I think if we are making recommendations for the future, even the not very distant future, it's clear that the cost of whole genome sequencing is going to be dropping into the range of $100 to $200. There was an announcement at so we wrote that somewhat prophetically in the white paper and in the recommendations. Then at AGBT, there was a first announcement by one group that said they were going to be releasing a sequencer whose reagent costs were $100. So that probably translates into something a little bit under $200 of total costs. And I think there's every reason to think that this will drop to $100. I think at $100 for whole genome sequencing, even though I've been one of the biggest proponents of whole exome sequencing for the purpose of getting as many samples as possible, I'm sort of now at the point of saying we really are in a world where we will cut over in the next several years the whole genomes. It will just become affordable. It's now reaching the point where it's equal to or less than the cost of collection and all of samples. And even in medical systems, for people who live 100 years just around, it's sort of on the order of $1 a year. So I'm biting the bullet myself, which is a big change for me in saying, I think we will cut over. So I think that's why we put it there. Obviously, it's something that individual funders and groups should decide for themselves. But I think the recommendation was to look that way and think that way, because that's sort of where it's probably going.
There are a few other scientific rationales. So fine mapping is much better serviced by having comprehensive capture of common genetic variation. This actually also improves diverse population capture. So a lot of the arrays are built off of what we know about different populations, and we know a lot more about European ancestry individuals than we know about other world populations, and so there's value there. It also helps for structural variation and some of the other kind of more complex variants. So I think there are good scientific motivations alongside the sort of forward-looking motivations that Eric just provided.
I think Carl is raising his hand. Carl Anderson.
Yeah, I was going to say, Carl, I see that you have your hand raised. I'm going to unmute, although we also did answer your question. Do you have anything you want to add, Carl?
Yes, I suppose.
We can hear you.
The others have. I suppose I'm less convinced that now is the right time to.
So, Carl, we're having a little bit of difficulty hearing you, but I think your question was sort of about the timing of this, and is now the right time to be making, how does timing factor into this decision?
So I could channel Carl, because I agree with his point on the scientific level. We talk a lot about this, which is, I guess there's two things that I would also add to this. One is there's whole genome sequencing for new biobanks in diverse parts of the world. This is essential now because we need to have a variation resource and an imputation resource and so forth that is comprehensive and deep in all populations. So I think there's no argument that we do that now. The question of what we invest in as a community for the deep dives into diseases, I think since those are already happening and are going to be funded by organizations interested in those diseases, that will be taken up on a disease by disease basis. The point being that we expect the crossing over point to be sometime in the not too distant future, but we will assess it as it comes.
Thank you. So there's quite an active sort of chat in the questions, in the question and answer about the importance of environmental information. Given the timing, I'm not going to sort of go into that in great detail, other than to say we're keeping track of these discussions, and certainly Rick and Richard and David, I think you guys really have given some really good feedback and considerations here, particularly about how to get the environmental information and what are the best ways to go about doing that. And so I think that's certainly something for ICDA to consider and think about as we move forward. I want to give sort of the last question for this session before we go back into the asking, coming from Herman DePazzo from Buenos Aires, who was really sort of asking about how ICDA can best support regions that are currently underrepresented in genetic studies, where science is also more difficult to carry forward because of resource limitations. And how can ICDA support these researchers, regions, groups, or communities to help properly develop the ICDA program and reach that sort of goal that that ICDA has scientifically? Cecilia?
So Nikki just answered, so maybe I think it's going to be covered in cluster 6 again, but Nikki, do you want to say something quickly to this as well?
Sure, I'm happy to do so. So recommendation 23 is specifically designed to push forward promoting global equity and to ensure that the work that goes forward is globally inclusive, both for participants and for researchers. So the ICDA is recommending active steps to decrease the barriers to research in some regions, which is exactly what you've raised in this point. And this can be through training, technology, and funding. So 3 different arms that we've identified where work needs to be done. It can be done working with funders to promote these global equity goals, as well as hands-on training, hackathons, online training, and then also securing equitable access to advanced technologies. So working to ensure that pricing is fairly kind of pitched to not exclude sectors of the research community. So I'm going to say all of that again in about 4 minutes time, but anyway, that's sort of the overview. Yeah.
Excellent. Thank you, Nikki. And on that, Cecilia and Eric, I will kick it back to you now. Cecilia, is when you can go into cluster 4.
Yes. So I'm going to progress into cluster 4 and ask Ben to talk us through the data-related recommendations.
Of course. Thank you, Cecilia. So there are 3 core data recommendations that we've kind of highlighted in the near term. The kind of first and perhaps most complex among those is the idea of a shared data platform. It is worth pausing here and noting that this is not a single centralized data set or sort of server where all of the data lives. Rather, it's a potential ecosystem of federated computational environments that will allow for different data sets to reside in different places, respecting the sort of ethical and compliance and regulatory considerations for each data set. But nevertheless, there's been a long tradition in human genetics of having these kinds of computational workflows readily available to many in the community. You can think of like the haplotype resource consortium as like a example of a online place where people can process data in a given way. So that is a sort of exemplar to guide our thinking about how we want to make sure that the ways to process and analyze genomic data sets can really be made accessible to as large a group as possible. Recommendation 11 really focuses on this idea of a kind of genotype by phenotype portal. So we have the GWAS catalog and the efforts that have come along with the GWAS catalog. There's an opportunity to kind of expand and enrich that kind of environment, as well as expand and enrich other kind of key environments and resources that are out there for the community. Things like Nomad and Bravo for allele frequency references, as well as the potential for thinking about other portals such as what GTEx is set up, or some of those other ways of accessing genomic information and results for the kind of wider research community is really the gist of recommendation 11. And then recommendation 12 really focuses on this idea of a handful of gold standard validation data sets. And that can kind of come in 2 different ways of thinking about. One is for kind of data processing. So you can imagine key data sets that if you can process this data set and get to the same answer, then you are now compatible with the standards that are being used more widely across the community. And that has obvious potential value for ensuring things like functional equivalence. And then on the other side, there are, this whole M to M to M challenge is focused on trying to get from the genetic mapping all the way through to the medicine. And so as we start to develop gold standard data sets for different pieces of that overall pipeline, those gold standard validation data sets should be made available as widely as possible, because they'll help us develop the methods and the approaches and the computational tools necessary to continue and expand this particular approach across as many different diseases as possible. Thank you, Cecilia.
Great, thank you. So after Ben, we're going to go over to Unnur Thorstensdottir, who is going to talk through cluster 5, which are recommendations 13 to 17 and represents the medicines group. Take it away, Unnur.
Can you hear me? Are you hearing me?
Yes.
Okay, good. Okay, so I'm part of the medicine group with Judy Cho and Mark McCarthy, and we have also worked closely with the Pharma Council, and Mark is actually on both in both groups. And we have 5 recommendations that are tailored towards the research community and also to pharma. And our first recommendation is the development of functional assays for systematic dissection of disease-related processes, for example, autophagy in ALS. And we have discussed whether this should be, focused on genetic pathways that cross disease boundaries, or whether it should be like to develop new platforms that are more customized to each disease. Then is the creation of tools to propel therapeutics development for high priority targets. And in this respect, we are talking about like development of antibodies, molecular inhibitors, and biomarkers that are very time consuming to generate, and for high priority targets, like both the research community and pharma could step in there. And we also see like a good avenue for this, the structural genetic consortium. That is actually is actually a good path forward for a number of these things that we need to develop. Then there is the development and to develop and apply new methods for discovering biomarkers of disease. We discussed this a lot, and there is sort of a new we are seeing new ways to do proteomics of plasma, and we discussed, and then there are like also proteomics of CFS, and there are a number of omics and other omics and imaging data that can be used as biomarkers. And in this respect, I think, for example, if we just talk about the obvious candidate plasma proteomics, then both like to use the plasma that has been collected through in large biobanks to actually do studies on plasma proteomics, as well as to incorporate this into clinical trials that would use this from the start, like phase 1, 2, and 3, to actually evaluate like the and target engagement and therapeutic response in clinical trials. Then we discussed a lot the how we can sort of use genomics to assist in clinical trials. And in that respect, we discussed a lot the polyclinic risk score that can actually increase the success of the trial based on selection into the trial, and it can actually reduce the number of adverse effects and the time to the enrollment. Like we could use even the known biobanks for that. So, and this is actually, there are some actually regulatory thinking that needs to be thought of, because whether this, how this would actually approve with regulatory authorities. And but this is actually a very important point that that can actually help and to reduce the cost of clinical trials, which is probably the most expensive part of the drug development. And then the last recommendation is sort of linked to this, is actually to ensure responsible use of the polyclinic risk score in medicine. So I'm done.
Perfect, thank you, Unnur. And for the participants, keep the questions coming in the side, and we'll address them when it's time for Q&A. And we're now going to go over to cluster 6, which we have already touched a bit upon, but that contains recommendations 18 and 23. And Nikki Tiffin is going to lead the discussion on those.
Thanks, Cecilia. Yeah, so very briefly, recommendation 18 talks about the policies for genetic studies, the data sharing and federated analysis. And it recognizes that we need clear policies and guidelines for data sharing and access. These have to be globally relevant, inclusive, and appropriate, also globally. And the idea is, or the recommendation is to collaborate with existing efforts. So we've flagged particularly GA4GH, which has been very active in this area. We certainly aren't recommending that we try and reinvent the wheel from scratch, but rather partner and consolidate what's out there in a way that is relevant across the board. So we've proposed a data compliance task force that would undertake this and could work with national authorities and funders, also bearing in mind that legislation varies globally. So keeping all of these in mind in building the guidelines. As I already touched on, informed consents and data permissions also need to be addressed in terms of guidelines for sound consent processes and the storage of permitted data uses and secondary uses, and also using standardized vocabularies for storing those permissions. We'd like to also recommend that we, in terms of people accessing data and researchers becoming data users, that we could standardize the user categories to facilitate access to appropriate data sets. So for example, if a researcher is labeled as being someone who's an epilepsy researcher, then they would be able to access data sets which have approved epilepsy secondary use. So by standardizing our vocabularies of how we store consents and data permissions, we can facilitate data use. And then policies for sharing data sets. There are different types of data sets that need clarity. So summary statistics and metadata are one such category. We need clear policies about when these are open and when they're controlled access summary data. Policies around data for strengthening genomic data resources. So this might be imputation panels, generating allele frequencies, or population level aggregate control data to have controlled data sets. Also policies and technologies for federated data analysis, that's also been touched on briefly. So this would be around the data sharing aspect of that. Community engagement guidelines are very important. These are going to, community engagement practices are going to vary globally. So these are guidelines to be able to be regionally and locally appropriate and to ensure inclusive policies. And to identify culturally accepted research practices. And then, and that's recognizing, as I already mentioned, that different cultural perceptions exist for work, for example, with cell lines, in vivo models, even blood samples can be culturally very differently approached. So community engagement guidelines can help to be sensitive to those. And then recommendation 23 is to promote global equity. And as I mentioned, this is about being globally inclusive, not only to increase our global array of participants, but also to include researchers. Active steps can be taken to decrease barriers to research for maybe under-resourced regions through training, providing technology, and for directing funding appropriately to those regions. So the ICDAbio recommends working with funders to promote that kind of approach and to promote global equity goals. And this could look like undergrad internships, could be postdoctoral fellows that are held in other parts of the world, and long-term projects, but then supportive researchers to go back to their home countries and undertake research there, and then also hands-on training and hackathons, and promoting the access to online training material to extend the reach of these kind of resources. And then, yeah, to sort of advocate for equitable access to more advanced technologies, genome sequencing, single cell characterization, computational analyses, and to try and advise on pricing, training, and platforms that can accommodate limited, things like limited bandwidth or technical capacity. So those are the main points. Thank you.
Great, thank you, Nikki. So then we have the last tranche of recommendations, 19 to 22, where me and Eric are on the hook. So Eric, do you want to start here?
Sure, I'm just coming over to this end. So
sure.
19 to 22 are meant to acknowledge really clearly that while many of the ICDA recommendations pertain to larger projects, creating resources, dealing with policies, things like that, it's really important to say that we don't know everything that's all of the scientific foundations, and it's really important to have individual investigator-initiated grants and small group projects that go off in entirely new directions to develop methodologies and technologies and new biological understandings of the genetic basis of common disease and training programs, because one can sometimes fall victim to thinking we know how to do everything right now. I think the other ICDA recommendations are built by looking at what's going on and trying to integrate and distill them. These are a clear statement to funders that lots of creativity is going to be needed. And we've tried to lay out those areas. And I think in some sense, this is the most straightforward one to implement, because funders have been doing this, and at least some funders have been doing this. And I think we're saying that doubling down on the importance of the individual creativity and the small collaboration creativity is going to be crucial to the success. I think ICDA would be glad to help funders in, if it's needed, to convene meetings about some of these topics, et cetera. But that's really the motivation for these recommendations.
Great. So then over to you, Caroline.
Okay, excellent, everybody. So again, a reminder, and for people who perhaps joined late, what we're doing now is a question and answer session with over 125 people on the call. We're not opening it to everyone just talking at once. So if you have a question, please type it in the question and answer box. You can also upvote other people's questions, or if you're not able to do that, you can also raise your hand. So before I go into a couple of the questions that have been coming in, and as hopefully as some people are typing some questions in, Ben, I have a question to you. It's a little bit more of a statement than a question. The data recommendations right now, sort of as written, focus on sort of the data resources, data platform need, and then not as much of a thing of sort of recommendation on the sort of data generators. And so I feel, and others have noted, that the recommendations could benefit from having more about sort of data being fair, machine readable, standards around that. There's a little bit of talking with GA4GH, data flow, et cetera. But do you sort of have some thoughts on where you see that in the current recommendations, or where that might be added going forward?
Sure, that's an excellent question, Carolyn. So the FAIR principles are, I think, perhaps more implicit rather than explicit in the data recommendations at this point in time. I think it makes sense to make them a lot more explicit. So, for example, there's FAIR includes interoperability, and we talk a lot about federated solutions, and for federated solutions to work appropriately, interoperability has to be a kind of core component of that. Similarly, with the kind of accessibility piece, a lot of the data recommendations really turn around trying to, in a sense, lower the bar for access, both in terms of how we think about accessing individual level data sets, but also how we think about accessing certain kind of core human genetic resources, such as imputation or even variant calling from raw sequence data as another way of thinking about accessibility. It is very much focused on what we do with the data and how we organize and coordinate and make available the data, rather than exactly what data is being generated. And the reason for that is that the kind of data generation activities are going to be much more naturally sort of built out in the scientific projects that ICDA is recommending. So, sort of where a flagship disease is in genetic mapping and where it needs to go in genetic mapping should be tailored to each individual flagship disease. And that's not really a kind of what we do with the data is not the same as what data sets we think we need to create and how we think about getting towards those data creation aspects. And so that was really the sort of remit and the way to kind of constrain the scope of the data conversation, because a lot of those kinds of core principles and approaches, I think, need to be shared so that we can really work together and go from maps to mechanisms to medicines. And I think that's that kind of going linearly through those things, particularly starting from that genetic mapping point of view and focusing and amplifying the importance of understanding genetic variation that is relevant to different diseases and how that works mechanistically is at least in my mind, a kind of major tenet and plank of the ICDA. Great,
thank you. I'm going to go next to a question in the question and answer box from Vijay Sankaran. And Nikki, I'm going to send this one to you. And it was really sort of following up on your discussion of data access. And can you sort of touch on how potential recontact could be facilitated in the context of genotype populations that are part of ICDA?
Sure, thank you. Yeah, so Vijay, I think from my perspective, probably the point that I would raise is that if informed consents are done appropriately, it's possible to get consent for recontact. And if the informed consents are properly stored, it should be fairly trivial to mine as to whether people are consenting to be recontacted or not. So I think it speaks to the idea of properly coding and storing consents. And I also can raise, and it maybe also speaks a little bit to Carl's question, that electronic health records can also be a great way to maintain contact with study participants. So if we can keep an open mind to working with electronic health records and provision of health care as a partner to the research enterprise, I think that certainly within Africa, that can be a very powerful way forward, and allow us sort of a more longitudinal follow-up of participants. So those are just a few ideas, but I'm happy to follow it up further.
Thank you. I'm going to back up now a little bit, Tuli. So this is a mechanism question coming in, even though we sort of moved past your section. And it sort of addresses the fact that there's a lot of focus on human genetics and phenotypes, occasional mentions of animal models. But there's also sort of gaps and holes in the overlap of animal-human data currently rendering translational work less effective. Do you have ideas on how ICDA could help to maximize the usable overlap between human and mouse work? And I guess a little bit related on that, can you sort of just talk a little bit more about how animal models, this is incorporating a question that came in online last night, could also be used to validate findings?
Yeah, we've had quite a bit of discussion about what would be the role of animal models in this work. And one of the challenges has been that the sort of like the mandate that we've had in the mechanisms working group is really sort of like have comprehensive, pretty sort of genome-wide, large-scale efforts, and some of the animal work is just kind of like, it doesn't have that kind of scalability that we have, like in our recommendations, try to avoid the sort of like single locus sort of approaches. But I think it's clear that animal models do have a very important role in this, and they need to be part of this effort. I think it's a fair comment that.
Tuli, we seem to have lost you.
Okay, can you hear me now? Yeah, sorry, I'm worried about my microphone settings. Yeah, so yeah, I think that's a good comment. I think that it's also something that maybe animal models come more into question when it comes to the medicines working group, since the cellular molecular mechanisms are in some ways something that we will be addressing by human cell line models and such efforts. I don't know if Unno or someone else wants to jump in when it comes to the medicines part.
Yes, can you hear me?
Yes.
Okay, yeah. Oh, yeah, we have discussed that, and you can actually set up with in animals, like a high throughput screening of, with CRISPR or any other methods. So we did discuss that, and I think that will be part of the sort of the functional assay to try to understand, the processes that a particular gene or genes are involved in.
So Uner, while we have you on, I wanted to follow up with a question. It relates to one of the questions that came in last night about recommendation 15. So as we sort of think about, there definitely is promise in the area of discovering biomarkers from disease, thinking about using machine learning methods, et cetera. But one of the questions is right now there's not really sort of systematic assessment of already known studied biomarkers. And so as you think about this discovery, can you also think about ways to think about systematic assessment with biomarker associations, or clearer discussion of what types of information or studies designs are needed really for going to biomarker discovery versus association? And how do you think one, particularly ICDA, might be able to contribute to such efforts?
Yeah, I think the genomics and the other omics are actually working quite well together. But you need a very complicated and machine learning mechanism to analyze all of these together. And I think that this is going to be actually critical now with the, for example, with the proteomics platform from SomaLogic measuring 5,000 proteins in plasma. It's just a new era of, and you can look at variants in the genome that affect the levels of protein, and you're getting closer to the disease. And I think that actually we will probably see in the coming years a lot of the biobanks that have gathered plasma now analyzing the plasma, and also this is going to be part of clinical trial, I believe. So you'll get a better understanding also from that area. I hope I answered your question.
Does anyone else want to add on that point?
I can add just quickly, also seeing Rory's comment that we, in the recommendation around clinical trials, I think we've sort of tried to acknowledge that we're not clinical trialists, and we have given a shout out to the Gates Foundation, Wellcome Trust, and also African Society sort of initiative around clinical trials, which Martin Landre, who's here in the BDI, where I'm in as well, is spearheading. So I think it's a good point, but to just build on to what Uner is saying, I think sort of we are aiming to liaise with other sort of forums and stakeholders here that are working on figuring out some of the aspects.
Do we lose Cecilia?
I think maybe her connection just
we seem to have lost
briefly. Yeah.
Maybe
Cecilia.
just a teeny bit
Did I freeze?
to the end of Cecilia's point for Rory's excellent question. I think
so Eric, I'm going to pause you for a minute for the people on the phone. So Rory's question, do you want to just summarize it in a sentence before you answer it?
So Rory's question was, well, it's wonderful that recommendation 16 thinks about how to make clinical trials smaller and more efficient, but maybe that's not a good idea because it might actually miss the fact that a treatment applies to a much larger population, or by doing a smaller trial, one might get a more limited evaluation of safety. So balancing the idea of is it really key to be doing the smaller trials? Maybe we should be finding ways to make it easier to do the larger trials. And I think Rory raises a really important question that the current version of the recommendations just doesn't go deeply enough into. I think it's really a complicated question, and we need to say more about it. There are certainly times when pharmaceutical companies want to do a small trial in order to get an approval in a limited indication, because then they'll get money for it, which means they could now run trials to expand the indication. So it might allow more trials to go on and more explorations to go on. On the other hand, the point Rory raises is absolutely right. It might not be the right thing. And I think we need to embrace the positives and potential negatives of this kind of an approach. And my guess is that it will be a mixed portfolio, and we're going to need to think about doing both. And as currently written, we don't acknowledge that. I think it's just a little bit too much on the small genomic trials are such a great thing side. And so I'm really glad that he's brought this up.
It's also, I think, worth emphasizing that this is really a series of exploratory discussions about how one might design that, what it might look like, and what some of the benefits and potential risks are. And I think we probably amplified the benefits a little bit more than risks. And I think Rory is quite rightly calling our attention to the risks piece. So this isn't like we have a clinical trial ready to go that we're going to do. This is more, how should we think about it? What are the potential upsides and downsides? And really, how would we do such a thing from a both scientific and regulatory point of view?
Yep.
So, sorry, and I got cut off, but I was going to say that I think it's an important question to raise, because a lot of people have talked about, PRSs and are they ready to go into clinic? And I think we tried to address that with, we should evaluate them in clinical trials to see if they are and what the potential risks are, which is the question I think we're really trying to address.
Right.
Yeah, I think we just need to keep our mind open for this opportunity or this avenue, and it doesn't maybe apply to all trials, but at least maybe to some.
Right.
Absolutely. And properly evaluate when it's working and when it's not going to work, and what are the optimal settings for it and what are suboptimal settings.
Totally agree.
So I'm going to change focus a little bit and sort of tie in one of the questions that came in online that was really sort of asking what are the opportunities to contribute to the white paper recommendations, and are there working groups, and who is membership open to? And I'm going to tie that also to some of the feedback that came in earlier about promoting training opportunity, or about training and thinking about the next generation of researchers. And one of the things that's discussed is promoting training opportunities, but I also wanted to sort of ask the panelists about what you think ICDA could do to promote early stage investigator involvement in ICDA itself. So two-part question there.
Well, maybe the first one about who's this open to have input into the white paper? The answer is everybody right now. I think the purpose of releasing a version 0.9 was to solicit input from the entire community. Anybody can be a member of ICDA just by signing up, and anybody can provide input into this. And so I think this has been an experiment, and I think we're going to try to keep this going, even when in the coming weeks we reach version 1.0. 1.0 isn't the end of the numbering system. I think there should be continuing opportunities for people to be raising questions to the working groups, to ICDA, and on a regular basis, we should be updating this. We could think, and it would be great if people would write with suggestions of what are the best ways to encourage people to do it. Bringing together additional small groups of people to do it, especially with regard to trainees, figuring out how to get people in their early stage career scientists to feel empowered to make these recommendations. I think what's come from the questions we've gotten already are that there's a lot of really good ideas, and that we're going to want, the point of ICDA is to bring together the wisdom of a community. So anybody who has ideas on how we can make that clearer to everybody, that this is open to all, and we're trying to distill all this input, we're very open to figuring out how to do that.
And just to add to that, so a group of us have already started to discuss what we can do in terms of mentorship schemes, about joint training programs. If we think about being a global resource, what's most helpful in different parts of the world? Because what's helpful for a training in the UK is most likely very different from somebody in India or South Africa or Brazil. So how do we make sure that we sort of capture that, so we don't do like a design of what helps somebody in the UK or the US, but more globally? So at every planning we try to sort of look at a range of diversities from stakeholders, gender, age, global representation and so forth. We can't always capture it perfectly, but it is at the core and forefront of our minds and hearts when we're planning this. So just want to emphasize that.
Great. Thanks all. So I do see that there are still some questions and comments that came in from Carl and Sasha and Loic. These seem to be more feedback than direct questions, and so we'll take those into account along with all the other feedback that's been coming in today as the group works. Oh, and a late breaking thing from Rory, which I haven't had a chance to read yet, but we will read and pay attention to. But in the interest of time, I'm going to send it now back to Eric and Cecilia to sort of close us out with some next steps and conclusions.
So do you want to go first, Eric, or should I go first?
I'll go quickly, and then I think you can close this out. I mostly want to observe that this experiment we just ran today, with more than 130 people joining in, just on this one town hall, we have an attendance sort of in the neighborhood of what the auditorium in Copenhagen was going to be able to hold, just a little bit more there. So I think this is already an example that there are ways we hadn't thought of for how to, on an ongoing basis, make sure that the whole community is engaged. We're going to go back and take the list of questions that people have laid out, the discussion on this call, and use this to rework 0.9 of the white paper into a 1.0, which I hope I'm sure we'll get done this month. But already I'm so convinced that the community feedback and this particular mechanism of a Zoom town hall webinar, which I never did before, is a great thing, and it points to how ICDA can do a better job than we'd thought about making sure it's a global community, distilling a path forward. I'll turn back to Cecilia.
Yeah, I want to echo that. I'm a pro webinar person now then. So I want to thank everyone. So I want to start to thank with all the working groups for the immense amount of work, and also the community for all the input that you have given over, we have tweeted out the recommendations, people have sent in recommendations to the ICDA office, or we have tried to sort of capture the discussion. We have really sort of tried to massage these recommendations with as much feedback as many of you as possible. And I think it's really sort of been an exciting progress over the last couple of months to get to where we are now. So I'm excited to move towards version 1.0. And it is wonderful to see that so many people are engaging. And the questions about how, somebody asked how we choose the disease projects, and I think Eric and I try to address that, saying that you should champion them. So if you have a disease you want to champion, start to think about what a flagship project could look like. And we're going to come back with more discussions about when it's appropriate, how to sort of move towards implementation. So that will obviously be the next step. But for now, focusing on the recommendation, it's been super valuable to get all your input. And also thank you to Caroline for hosting the discussion so well today. So I think we should close out here. And for those of you who want to, there is a second round of town hall in the afternoon. If you've now increased your appetite, then it will probably run even smoother now that we know how to do. We're going to be here, so we're looking forward to discuss with as many as possible then as well. So thank you, everyone, also for calling in and giving us your time and your thoughts.
Thank you.
Thank you.
All right. So we shall adjourn for now, and we're back in, I don't know.
3 hours.
3 hours
Yeah.
for round 2.
Yeah.
Thank you, everybody.
Thank you. Over and out.
Bye.